Thank you. Well, it's a great pleasure to be here and participate in the symposium on how neurons and circuits of neurons compute. And the question that's always interested me most about neural circuits is how they use their plasticity to tune the way their computational performance. And here I'm using the term compute to mean transform an input into an output. So my lab has focused on the question of what are the algorithms that neural circuits use to tune their performance in this way? What are the rules? And we know that individual synapses don't know what's happening in the world, but they're making a local decision about whether to change or stay the same based on the pattern of input they experience. So my lab is interested in better understanding the local rules that synapses use to make these decisions, and ultimately how those decisions are coordinated or at least work together across a circuit to define an algorithm for tuning the circuit's performance. Now, we have some clues about this from the work that's been done over many years in brain slices, where we know, for example, about Hebbian plasticity, whereby cells that fire together tend to wire together. Coincident activity in a pre- and postsynaptic neuron can induce changes in the synapse connecting that neuron, those two neurons. And we also know about spike timing-dependent plasticity, which can alter synapses in opposite directions, potentiation or depression, depending on whether the presynaptic neuron fires just before or after the postsynaptic neuron. And these two general rules, these two ideas about plasticity, Hebbian and spike timing-dependent plasticity, have shaped a lot of the work on learning and memory, including the work in my lab. But today I want to focus on some of the ways in which these rules fail to fully account for what's happening in a neural circuit as it learns. And I would go so far as to claim that even if we knew the activity of every single neuron in the relevant circuit during some experience, at this point we'd be hard-pressed to predict, using these rules, which synapses would change and which would not. So in response to Mark's question of when do we know we're there, I wouldn't say we're there if we got to that point, but that would be an important milestone that would then let us go to ask, well, what is the logic of those rules? If we understood the rules in order to be able to predict which synapses would change, then we could at least be ready to go to the next step. So that's kind of the goal, the big vision. Today I'll tell you about some progress we're making towards this goal. And to make this general problem tractable, we've turned to this brain area you heard about in the first talk by Angus Silver, the cerebellum. And I could spend hours waxing poetic about the advantages of the cerebellum for studying learning, but I will restrain myself and just say that the circuit architecture is relatively simple. And we know a lot about how neurons in the cerebellum encode information, at least in the parts of the cerebellum that I'll talk about. And this makes it possible to try to link events happening in the world with the patterns of activation in the circuit and the changes that are occurring at synapses, and connect the dots between those different levels. Now, I'll focus today on the rules, some progress we've made in understanding the rules controlling the recruitment of an associative form of long-term depression at parallel fiber to Purkinje cell synapses in the cerebellum. And almost 50 years ago, David Marr and James Albus hypothesized that these synapses are a key site of plasticity during cerebellar learning. So this model still influences a lot of the work in the field today, so I'll take you very briefly through it. There are two main inputs to the cerebellum. You already heard a lot about the mossy fiber input pathway this morning. Mossy fibers synapse on granule cells that make the parallel fibers, which provide excitatory input to the Purkinje cells. The second input is from the climbing fibers, which arise in the inferior olive and synapse directly on the Purkinje cells as well. And the model is that the Purkinje cells, which are the sole output neurons of the cerebellar cortex, that Purkinje cells represent all of the different responses you might want to make, whereas the parallel fibers represent all of the different contexts in which you might want to make those responses. So by modifying the appropriate set of parallel fiber to Purkinje cell synapses, you could learn to make the right response to a given context. And if the coding in the parallel fibers is sparse, as Angus Silver described this morning, then this would let you do learning that's highly context dependent, which is the kind of thing we think of the cerebellum as doing for fine control of movement, for example. Now, the model further hypothesized that the instructive signals controlling plasticity at the parallel fiber synapses were provided by the climbing fibers. Marr and Albus actually differed as to the nature of these instructive signals. Marr thought they were reinforcing signals that would strengthen synapses that led to an appropriate response, whereas Albus thought they were error signals that would weaken synapses that led to an inappropriate response. And it was Albus's error-correcting version of the story that's best supported by the subsequent physiology that's been done over the years. If you record from climbing fibers during a variety of behavioral tasks, they seem to encode errors. And in brain slices, activation of the climbing fibers can trigger long-term depression in the parallel fibers that are active simultaneously. So this is an associative form of depression that we describe as anti-Hebbian. Okay, so this morning I want to talk about some of the progress we've made in understanding the rules controlling the recruitment of long-term depression during cerebellum-dependent learning. And this work has progressed by taking advantage of the behavior shown here, the vestibulo-ocular reflex, or VOR. I'm very proud of my performance as the R on our recent lab ski trip. The VOR is a reflexive eye movement driven by vestibular signals. When your head turns, this movement is sensed by the vestibular system, which drives an eye movement in the opposite direction. So if your head turns left, your eyes turn right, and vice versa. And this reflex tends to stabilize the direction that your eyes are pointing in space as your head moves about in the world, and this improves vision by stabilizing images on the retina. Now, in order to do its job of stabilizing images, this reflex needs to be well calibrated, and we study the form of cerebellum-dependent learning that performs this calibration. If for any reason images are not stable during head motion, then this cerebellum-dependent learning kicks in and can adjust the amplitude or sometimes the timing or dynamics of the eye movements until images are again stable. Normally, this might occur during things like changes in the strength of the eye muscles during development or aging. When you get a new pair of glasses with different magnification, you need to adjust your VOR. But in the lab, we can produce big changes over short time scales by pairing a vestibular stimulus, head motion, that we control with motion of a visual stimulus that we also control. And depending on the relative directions of head motion and image motion, we can induce either an increase or a decrease in the amplitude of the eye movement response driven by the VOR. So if we move the visual stimulus in the opposite direction from the head, then the eye movements need to be bigger than normal to keep the image stable on the retina. And so over the course of many such pairings of vestibular stimulus with oppositely directed visual stimulus, there's a gradual learned increase in the eye movement response to that same vestibular input. If instead we move the visual stimulus in the same direction as the head, then the eye movements normally driven by the VOR would need to be suppressed to keep images stable on the retina. And so over the course of pairings of vestibular stimulus with visual stimulus in the same direction, we see a gradual reduction in the eye movement response. Both of these forms of learning depend on the same part of the cerebellum called the flocculus. Okay, so now I'm going to map this VOR learning task onto the Marr-Ito model that I described. We know that the vestibular, so we're pairing a vestibular stimulus with a visual stimulus. The vestibular input is encoded by the parallel fibers. The parallel fibers excite the Purkinje cells, which drive eye movements. The goal of the reflex is to stabilize images. So image motion on the retina represents what we call retinal slip, represents an error. And that error is encoded by the climbing fibers that are driven by this image motion. Okay, so here's how that looks when we're actually doing the experiments. So we turn the head left. This is, say, angular velocity of the head. We turn the head left, right. That drives an eye movement in the opposite direction, right, left. To induce an increase in the VOR, we move a visual stimulus exactly opposite to the head. This drives a response in the climbing fibers. And if instead we want to induce a learned decrease in the VOR, we move the visual stimulus exactly with the head, and that also elicits a response in the climbing fibers. And the difference between these two training paradigms is whether the on direction for the climbing fibers, the direction that makes the climbing fibers increase their firing, is happening during head motion in one direction, which is ipsiversive toward the side of recording for VOR increased learning, and the on direction for the climbing fibers happens during a contraversive head movement during VOR decreased learning. In both cases, if you follow, so the same climbing fibers encode the errors during both kinds of VOR learning. If you follow Purkinje cells through learning, their responses change during learning, and they change so that their responses get progressively smaller during the stimulus in the direction that made the climbing fibers fire. And that's true for both of these. So we see changes that are in the Purkinje cells in the direction that you would expect if the climbing fibers are inducing a depression of their excitatory parallel fiber inputs. So none of this is controversial. These happen to be recordings from climbing fibers and Purkinje cells in my lab, but it had been shown previously by a number of different labs. The results are incredibly consistent. And I would add that these responses of the Purkinje cell drive an eye movement that is, in each case, more controversive or less ipsiversive, which, depending on whether it's summing or inhibiting the normal VOR response, can give you either an increase or a decrease in the amplitude. Okay, so this all makes sense. It could plausibly be explained by climbing fiber-triggered depression of the excitatory inputs. It looks like LTD, it smells like LTD, but the controversy that raged in the field for decades is whether these observations actually reflect the induction of LTD, if LTD is the mechanism for these changes observed in the Purkinje cells and the eye movements. And so I'll show you, in the first part of the talk, I'll show you recording, stimulation, and perturbation experiments we've done in my lab to try to resolve this. Using the three main approaches available in systems neuroscience, we've obtained convergent evidence suggesting that under these conditions, LTD does seem to be the explanation for these observed changes. But in this kind of training, it does not. So the recording approach was conducted by Hannah Payne, a graduate student in the lab. And she took advantage of variability in the climbing fiber responses to ask how tight is the correlation between the occurrence of an error signal in the climbing fiber and the induction of changes in the circuit. So I showed you histograms. The climbing fibers on average respond when there's an error, but individual climbing fibers respond on some trials and don't respond on other trials, even when the error is present. And so you might expect that if the climbing fibers are what's driving the plasticity, then on the trials where the climbing fiber fires, you'd get a little bit of plasticity in the circuit. On the trials where it doesn't fire, you wouldn't get plasticity. And then it fires again, you get a little bit more. And this is what Javier Medina and Steve Lisberger in 2008 very beautifully showed seemed to actually occur during another form of cerebellum-dependent learning, smooth they showed that they saw this tight correlation between the occurrence of a spike in the climbing fiber and the induction of plasticity. So we did similar experiments, and the way this works is with an extracellular electrode, we can record two kinds of spikes in Purkinje cells. Simple spikes, which are normal sodium spikes that go at about 50 or 100 hertz spontaneously, and complex spikes, which are a reflection of the climbing fiber input to a Purkinje cell. Cell gets input from a single climbing fiber, and each spike in the climbing fiber produces a big calcium spike, which we record on the extracellular electrode as this complex spike. So we get two signals for one. We can see the climbing fiber input to the cell and how the cell's output changes. So we look at pairs of trials, and we choose pairs where the climbing fiber fired on the first trial and ask how the simple spike output changes on the Is there a reduction in Purkinje cell output, as you might expect if the climbing fiber spike induces long-term depression? And then we compare that with trials where the climbing fiber didn't fire. So we do this during VOR learning in the increased direction, and we saw a very clear change in Purkinje cell firing. If the climbing fiber fired on the first trial of the pair, then there was a big reduction in firing on the next trial. This is in the direction you would predict from LTD. This is similar to what Medina and Lisberger had reported for smooth pursuit learning. So that all fit the model. In The controls, the pairs of trials where the climbing fiber didn't fire, we saw no change in the Purkinje cells output on the next trial. So this trial-to-trial change was not an inevitable effect from giving them some kind of training, but rather these changes seem to be happening in tight coordination with the occurrence of spikes in the climbing fibers. Okay, so all that fits the LTD model, but if we do the very same analysis in the very same cells during VOR decreased learning, so now we just change the training we're delivering, do the very same analysis, we got totally different results. So now we see no change. If the climbing fiber fires, we see no change in Purkinje cell output on the next trial. And remember, these are actually the cells I showed you in this slide. So the climbing fibers are encoding errors during both kinds of training. The Purkinje cells, if you follow them across many trials, are decreasing their firing rate during the stimuli that made the climbing fibers fire. But in this case, these changes seem to be happening in a way that's tightly correlated with the occurrence of these error signals in the climbing fiber, and in this case, they do not. Okay. So that suggests that this climbing fiber triggered LTD is contributing to learning in one case, but not the other. But that's just one correlation kind of approach. We also looked at stimulation. So this is work from postdoc Ria Kimpo and graduate student Jacob Rinaldi. They said to the extent that climbing fiber triggered LTD or other plasticity in the circuit is contributing to VOR learning, we should be able to bypass the sensory feedback about errors and directly stimulate the climbing fibers optogenetically to roughly mimic how they normally encode errors during learning and induce behavioral changes. And this is facilitated because the coding of the image motion by the climbing fibers is incredibly uniform. The vast majority respond to image motion away from the side on which you're recording. So that allowed them to go in and try to roughly mimic their responses during learning and see if they could bypass the sensory feedback and directly induce changes in the behavior. So for these experiments, they use sinusoidal stimuli, so the head turns left-right. The visual stimulus, if we want to increase the VOR, moves in the opposite direction, and that makes the climbing fibers fire during the ipsiversive part of the vestibular stimulus. If we want to decrease the VOR, we move the visual stimulus with the head. That makes the climbing fibers fire during contraversive head motion. So Ria and Jacob asked, can we just get rid of the visual stimulus, stimulate the climbing fibers at the time they normally fire, and can we induce VOR learning in this very direct way? As a control, so the way we do it is test the VOR, pair the vestibular stimulus with climbing fiber activation, and then test in the absence of the activation. As a control, we just omitted the activation of the climbing fibers. This produces a non-associative habituation of the VOR response over 30 minutes. But what happens if we stimulate the climbing fibers at the time they normally fire during training to make the VOR bigger? We saw a very robust increase in the VOR relative to pre-training and relative to the control condition where we didn't stimulate. And this lasted at least 2 hours. After 2 hours, the non-associative habituation decays, but we still see this very robust associative effect of stimulating the climbing fibers. Okay, what happens if we stimulate at the time that the climbing fibers normally fire during VOR decrease training? If we paired with a visual stimulus, we can induce a decrease in the VOR well below this habituation control. But when we stimulate the climbing fibers to try to make that happen, we did not get a decrease below the vestibular-only control. In fact, a trend towards an increase. And I'm showing just one kind of stimulation paradigm we used, which had 3 stimuli. We tried less, we tried more, we tried everything we could think of. I can't rule out that there is no way to stimulate the climbing fibers and make the VOR decrease, but we tried a lot and we didn't find it. Okay, so that suggests that the climbing fibers are not carrying the error signals that drive VOR decrease learning. That was from the recording and the stimulation experiments. We also took a perturbation approach, and Ed Boyden was the first one to do this way back when he was a graduate student. He asked what happens if we disrupt LTD. make a molecular genetic manipulation that disrupts LTD. And the one that Ed looked at, that we have now since looked at other lines, but Ed looked at the CAMK4 knockout mice. These mice, CAM kinase 4 is a kinase that phosphorylates Krebs, which has been implicated in the late phase of plasticity in many systems. These are results from David Linden's lab showing that CAM kinase 4 is important for the late phase of cerebellar LTD. So normally a pairing produces a long-lasting depression that lasts as long as they can hold the cell. But in these knockouts, there's an initial depression that then decays back up to baseline. So missing the late phase of LTD, Ed asked what VOR learning looked like in these mice, and he found that the acquisition of learning in both directions was normal. The blue the blue is the knockout, black is wild type, totally indistinguishable. But then if he put the mice back in their home cage and came back 24 hours later and tested retention, then he saw that the retention of the learned increase in the VOR was significantly impaired in the CAMK4 knockouts. Now, surprisingly to us at the time, because we were thinking VOR increase and decrease learning was just a matter of LTD on different subsets of parallel fibers, so we were very surprised to find absolutely no difference in the retention of the learned decrease in the VOR. So this told us that whatever was supporting learning in the two directions, different mechanism was supporting VOR increased learning and suggesting that LTD contributed selectively to learning in that direction. So this suggested a highly selective contribution of LTD to learning in one direction, not the other. But Ed also found selectivity in another dimension that was very striking. So the results I showed you so far were done with sinusoidal training and testing stimuli at a frequency of 1 Hertz. Head turns back and forth at 1 Hertz. Visual stimulus moves with or opposite. We got a very similar result if we went to a higher frequency of 2 Hertz, impairment of the learned increase in the VOR. But if we went to just a slightly lower frequency, the phenotype went away entirely. So I think this was a really important observation that Ed made, that very subtle changes in the way that you do the training or testing can determine whether you see a phenotype or not. This is a change from 1 Hertz to 0.66 Hertz. We actually did 1 and 0.5, and the reviewers made us go back and do 2 and something in between 0.5 and 1. So you can see that there's this very sensitive window where the behavior goes from being LTD dependent to not. And I suspect that this could be the case. So I'm often asked, well, is the mechanism of VOR learning the same as eye blink conditioning or rotor rod learning? And my answer is always, well, it really depends. The mechanism of VOR learning is not necessarily the same mechanism as VOR increased learning. That there seem to be multiple mechanisms in the circuit that can produce a similar behavioral outcome, and which mechanism gets recruited is highly dependent on how you train and test. So there's a lot of talk now about how results from mice fail to translate to the clinics. I tend to think that I wouldn't put all of the blame on the mice. I think it might really be, in large part, our mistake in the past of overgeneralizing from one test of cerebellum-dependent learning to cerebellum-dependent learning in general, or from one test of amygdala-dependent learning to fear in general. So I think we need to be really careful about not overgeneralizing our results. And here's another example. So that was one. This is another line of mice that we've looked at more recently. This is under review. That also had perturbation of LTD and showed the exact same phenotype that Ed had seen, highly selective perturbation in these mice with disrupted LTD of VOR increased learning at high frequency and not VOR decreased learning. Now, and if I look at other labs, Chris DeZeo's lab in Holland has done a lot of work on this, and sometimes he's reported LTD deficient mice have VOR learning deficits and sometimes not, and that also seems to fall out along the lines of whether he tested at frequencies above 1 Hertz or below 1 Hertz. Now, you might imagine that this is just reflecting the fact that something about VOR increased learning at high frequencies is just harder than the other kinds of VOR learning. And whatever you do to perturb the circuit, you might expect that this would be the first thing to go. That's not what we find. So I just show one example here. We've looked at mutants of the PQ calcium channels, and they show a very different pattern. So they show sparing of VOR increased learning at high frequencies, but impairment of low frequency VOR decreased learning, impairment of the ability to modify the phase of their VOR. So what we're finding is that different subsets of mechanisms are recruited by different kinds of VOR learning. And you really have to be specific about how you're training and testing if you want to get the full picture of that mechanism's contribution. Okay. Just to step back a little bit and summarize this first part, we've known since the work of people like Brenda Milner in the 50s that there are different kinds of learning and memory that depend on different brain areas. But I think most of us have been hoping that when you look within a given brain area, that the question of how that brain area learns would reduce to something simple, like consistent recruitment of a particular mechanism, cerebellar LTD, or maybe a small set of mechanisms. But the hope was that the same thing would happen in that circuit every time it learned. Our results from cerebellum-dependent VOR learning suggest that that's not the case. There are different mechanisms that can be recruited in different combinations depending on how you do the training. So my lab is working now to try to define what is the taxonomy of cerebellum-dependent learning, how many different components are there, and better understand the rules determining when each mechanism is recruited. Okay, so the first part of the talk, I provided evidence that cerebellar learning may or may not recruit this LTD mechanism that's the focus of most textbooks. In the second part of the talk, I'm going to switch gears and show you recent unpublished data suggesting that the rules for induction of LTD at these parallel fiber to Purkinje cell synapses can vary across different parts of the cerebellum. That the same anatomically defined type of synapse can have quite different rules, be obeying very different rules for plasticity. And this is something we kind of stumbled upon because we were worrying about how the cerebellum solved this problem that the error signals that drive the learning are delayed. So parallel fibers activate the Purkinje cells, which produce some response. And by the time the feedback about that error comes back to the cerebellum, there's been quite a delay between the activation of the parallel fibers and the activation of the climbing fibers. So how can the circuit deal with these delays using what we think of as coincidence-based plasticity? This is what theorists call the temporal credit assignment problem. So one solution is suggested by what had been shown about cerebellar LTD previously, which is that there seemed to be a pretty broad window for its induction. You can get LTD with simultaneous activation of parallel fibers and climbing fibers. You can get it when the climbing fiber is delayed 100 or even 200 milliseconds. You can get it when the climbing fiber comes before the parallel fibers. And I'm showing these results from Wade Regere's lab because he makes this nice curve. But across the field, many different labs have used quite a broad range of parallel fiber, climbing fiber, stimulus protocols, and shown robust LTD. So one solution for the delay in the climbing fibers would be to have pretty broad tolerance limits for induction of the plasticity. But this didn't sit right with me because what we know about cerebellum-dependent plasticity is that it supports these incredibly precise behaviors. We can learn to time our movements to within less than 10 milliseconds, and I had a hard time believing that that kind of incredible behavioral precision could be supported by a mechanism that seemed so sloppy. So we think we found a resolution for this mismatch. This is just to show evidence that in vivo, in the VOR circuit, there does seem to be compensation for this delay that's quite precise. So this comes from that trial-by-trial analysis that I described earlier. And what I had shown you before were results when we aligned on the trial start, which is what Medina and Lisberger had done previously. But we reanalyzed the data, aligning not on trial start, but on the time of the climbing fiber spike. So if you look at the time of the changes in Purkinje cell firing relative to the time in the trial when the climbing fiber spiked on the last trial, we saw that there was no decrease in Purkinje cell firing at the time that the climbing fiber came in, but there was robust change in Purkinje cell firing about 120 to 150 milliseconds earlier. And this is very interesting because the delay for the visual climbing fiber response is on the order of 120 milliseconds. So it seems like the plasticity that's occurring in vivo does not alter synapses active at the time that the error signal from the climbing fiber got to the cerebellum, but at the earlier time when the error was actually being generated. So in vivo, it looked like the cerebellar circuit was somehow solving this temporal credit assignment problem. And we wanted to know what was the synaptic mechanism for this. Just the mismatch. Here's what we'd seen at the synaptic level, and here's what we needed to account for what was happening in the intact circuit. So a very talented postdoc, Aparna Suvarthan, has found what looks like a solution to this problem. And she did this by comparing the rules for LTD in different parts of the cerebellum. Virtually everything we thought we knew about the synaptic physiology of the cerebellum had previously come from the vermis, because it's easy to slice. But Aparna wanted to study synaptic plasticity in the flocculus, which is the part that supports VOR learning. So she made slices, brain slices from the two parts of the cerebellum. And she started by using kind of a typical LTD protocol in the cerebellum, which is simultaneously activate parallel fibers and climbing fibers, do that 300 times. And if she does that in the vermis, she got exactly the same results everybody else had reported, which was robust LTD. If she did the same thing in the flocculus, she did not get LTD. She got a very robust LTP. And this is, in the cerebellum, the LTP is non-associative. This is what you get with parallel fiber alone. So the very same pattern of activation yielded opposite changes in parallel fiber to Purkinje cell synapses measured in the vermis versus the flocculus. We had assumed everything we knew from the vermis would apply to other parts of the cerebellum, and that seems to have been a bad generalization because there seems to be very substantial regional variation in the plasticity rules for the same anatomically defined type of synapse. So we already knew, we had the evidence I showed you that LTD seemed to be contributing to the VOR, so we felt like these synapses should be capable of LTD, and we were wondering why we weren't seeing it, and we thought about, whoops, this delay that I mentioned, that when there's image motion on the retina, which is the error, it takes about 122 milliseconds for that error signal to reach the cerebellum on the climbing fibers. And this 122 millisecond number had come, it was something I estimated when I was a postdoc in Steve Lisberger's lab by using many different stimulus frequencies and then looking at the phase shifts. Okay, so we thought about this delay and tested the floccular parallel fiber to Purkinje cell synapses using this biologically relevant delay in the climbing fiber activation, and then we saw very robust LTD in the flocculus. We found that this tuning for the behaviorally relevant delay was incredibly precise. So we saw LTD if we delayed the climbing fiber by 120 milliseconds. If we delayed by only 100, we got LTP. If we delayed by 150, we got nothing significant, but certainly not depression. Now, for the students in the room, I should tell you that the way I described the experiments is not how they were done. We tested 0, we got no LTD. We tested 100 because it was about 120. We got no LTD. We went to 150, we got nothing. Postdoc pulling her hair out, I'm pulling my hair out. And then finally, out of desperation, we said, well, why don't we try 122? We tried 120, and there it popped out. Persistence. Stubbornness. Okay, so it's highly tuned. And this is unlike, this form of plasticity that we found here at the parallel fiber synapses in the flocculus is unlike plasticity that I know of at any other synapse. It's similar to spike timing dependent plasticity that's been described at many synapses in that it has a very tight window of tens of milliseconds for the two neurons to be activated. But it's unlike STDP in that STDP those windows are centered very close to coincident, whereas in the flocculus, it's shifted over 100 milliseconds from coincident. So it's precisely timed to an interval that's over 100 milliseconds. This LTD in the flocculus also, just to put it in perspective, compared to what we had seen previously in the vermis and others had seen. So to just compare these are on different time scales. This is the plot I showed you before from the vermis. Here's the flocculus. If I just expand this out to be on the same time scale, there's the difference. Okay, so we found this incredible temporal precision that matched what many people had seen at the behavioral level, but what was different than what had been seen in the vermis. We had a hard time believing, though, that the LTD in the flocculus is temporally precise and the LTD in the vermis is not. So we went back and looked again to see if we could find evidence for temporal precision in the vermis. And to do this, we used a shorter form of plasticity so that we could look at the tuning of individual neurons. So analogous to the trial-to-trial plasticity that I showed you in vivo, we looked for the effects of single pairings in a slice. So we tested the parallel fiber synapse. We did one pairing with the climbing fiber, and then we tested again. And so we just used this ratio of the change in the EPSP after one climbing fiber spike, like we had seen in vivo, that induced changes. We found in the slice that this also induced changes. And in the flocculus, this single trial plasticity or short-term plasticity was tuned for the same interval between parallel fiber and climbing fiber as we had seen for the long-term depression. So highly specific for the 120 millisecond interval. Then we went to the vermis, and we found that coincident activation gave us this robust single trial depression. We also got it with a 100 millisecond delay of the climbing fiber, 120, 150. It depended on parallel fiber climbing fiber pairing, but it didn't, like the LTD that had been described before, it didn't seem to have temporal specificity when we averaged across the population in this way. And that was the important bit, because when we then looked at individual cells, we found that the cells that depressed for coincident parallel fiber and climbing fiber activation showed very little depression for the other intervals. And likewise, the cells that depressed robustly in response to a 100 millisecond delay showed little response to the other delays. And so on, we found some that were tuned for 120 and some that were tuned for 150. So although if you just average these all together, you get something that looks like it doesn't have temporal specificity. That seems to be coming from averaging across a heterogeneous population of cells, each of which is tuned to a specific interval. Okay, so we have this model. I mean, before Aparna started doing this work, we really thought, that LTD was LTD was LTD. But what Aparna is finding is that the rules for plasticity at different parallel fiber to Purkinje cell synapses seem to vary. And we would speculate that in the vermis, like in the flocculus, the cells might be tuned to the behaviorally relevant delay. In the flocculus, it was easy to show that because the climbing fibers have such uniform responses. We know what the relevant delay is. The vermis is much more, has diverse functions that have been described. We know there are climbing fibers that come from the spinal cords and reflect somatosensory inputs at very short delays. In these same two lobules in the vermis that Aparna recorded, imaging studies have reported cognitive responses that you would imagine would have much longer delays. So it's interesting to speculate that the tuning for different intervals may reflect the tuning of cells for their climbing fiber delay. Okay, so to summarize, I told you that synaptic plasticity mechanisms are not as general purpose as we had previously thought, that different mechanisms are recruited within the same circuit during learning, depending on how you do the training, and that the plasticity rules at an anatomically uniform population of synapses can vary in a way that seems to reflect their function. I already recognized most of the key players, and so I'll stop there and take questions.